Hi, everyone. Welcome to Foresight's Molecular Machines Group. I'm super excited to have everyone here again. Today, we are Zihao Ou discussing achieving optical transparency in live animals with absorbing molecules. I will share a lot more about you in the chat, but it's a real pleasure to have you on. Thanks, everyone, for joining, and the stage is yours.
Thank you, Alison. Yep. Let me share my slide. Okay. I believe it should be available to everyone. So I changed the title a little because to attract more people. So today I'm going to share with you a story about how do we get closer to the invisible man. A technical more terminology is how do we change the tissue transparency for non-invasive imaging. So my name is Zihao. So I'm an assistant professor in the physics department at University of Texas at Dallas. Usually people get confused or curious, what are you doing in physics department and why are you doing biology in physics department? And today, hopefully you can see that just through my talk, how can we use physics to help addressing some of the biggest challenges in biomedical field. So what you're seeing here is actually we have a beaker that we are extracting some of the food dyes from these snacks. So today I'm going to share with you how these molecules in the snacks can actually help us tuning the optical properties of tissue. So we are making progress towards the invisible. So I would like to start this introduction, especially to many senior audience that I guess you're familiar with the topic of Invisible Man, which is a novel that was first released in 1897. It is such a successful novel that it has been adapted to various of different versions of movies. Hundreds of years later, when I read this again, I get pretty excited to see how actually a non-scientist describes this process. So I would like to just bring your attention to this. A scientist who has devoted himself to research into optics and find a way to change the body's refractive index to that of air so that it neither absorbs nor reflects light. And you can see that it actually starts to talk about one of the key concepts, which is refractive index. Hundreds of years later, it is still the key concept that I'm going to talk about today, but I'm going to share with you what we have learned and what we are doing, which part is correct, which part is wrong in terms of his original design. So I usually like to start with one simple example, a live example of bubbled water to share with you some physical insight of refractive index. So why some tissue or optical system are not transparent to light? It's usually not because it's the material itself, it absorbs light. So for example, in this example, we have bubbled water. It's a very simple system. You have air and water. So if you think about these two components inside the system, air is transparent, water is transparent. None of them should interact with light. However, when you shake them and they form a lot of optical interfaces and it suddenly become opaque. So I see the key concept here is that it doesn't mean that individual components need to be blocking the light, but when you mix materials with different optical properties, it will start to block the light. So more straightforward way everybody knows is when light goes through from air to water, it will bend and it will change its propagation direction. So very similar in biological tissue. Every time it hits material with different optical properties, it will change its propagation direction. So when I see these optical properties, these are basically what we call refractive index. So this is a simple schematic, but in real life, this is much more complicated. It's still a schematic, but as we can see, as we know that inside biological tissue, most of us are made of water. So 70–80% of us are water, and water has a relatively low refractive index, about 1.33. When inside this aqueous environment, we have a large number of functional molecules, cell nucleus, organelles, lipids, fibrils. They typically have a higher refractive index. So if you draw a line across this cell, what you will see is that your refractive index is not a straight line. It will go from low, high, low, high. And all this kind of refractive index contrast is causing the scattering of light, which is very similar to the example I gave at the beginning, a bubbled water. So why our tissue has such all have different optical properties? This is not necessarily a bad thing because a light is one of the key method how we actually interact with the environment. For example, in our eye is like a very sophisticated device how we actually interact with the external environment. Part of our eye actually can focus the light, just like a lens. Part of our eye can actually guide the propagation of light, like optical fiber. And there are also cells that contain certain molecules can filter the light, just like to increase the sensitivity at the detector site. So if we think about all these kind of microscale structures, how does the light nature actually achieve this? These are all achieved through this complicated design of refractive index. So it's not necessarily a bad thing. It all depends on what we actually think about this refractive index. But in a lot of cases, because it strongly interacts with light, it's not good for the biomedical imaging. It limits the penetration depth of optical penetration. And usually this is a combination of two effects. One is absorption, the other is scattering. So here, what we're showing is two examples. One is absorption dominated, when the light tissue absorption is much stronger than the scattering. And the other one is scattering dominated, when the scattering is much more stronger than absorption. In these two scenarios, you can see when the absorption dominates, we can see light propagates into the tissue and gradually terminate after certain lengths. And in a scattering dominated case, instead of just propagate follow this path, what you will see is the light gets scattered into a much larger volume, which is not able to propagate further. It's not because of it is absorbed. It's actually because it's scattered into a much larger area or volume. So usually what we think is which one of these cases is more dominant. If you have a laser pointer, something that usually when I do in-person talk, I'll just use a laser pointer and shine onto my finger. What you see is that my whole finger will actually get illuminated. This is more close to the scattering dominated example. So these are cases when the tissue doesn't have much absorbing molecules. Usually scattering is about 10 to 100 times larger than the absorption coefficient if you use a quantity. However, this is not necessary for all the tissues. Another example is if you are dealing with blood, where we have hemoglobin, which absorbs strongly in certain wavelengths, absorption can be dominating. Yep. So, right. So because of this strong scattering inside the tissue, light will shine onto this tissue. We will see that it's scattered into all different directions. From the refractive index perspective, this is due to the fact that we have all different kind of refractive index contrast. So if we can reduce this refractive index contrast, we can make the tissue transparent, or light can go through without much scattering. So, but how can we actually achieve that? So in the past, people have been discussing ways, for example, we can change the refractive index of water to some of the high refractive index components, or you can remove some of the high refractive index components so that everything has a relatively low refractive index. So from a physics perspective, I started to think, is there a way that I can tune the refractive index of certain material without removing any components? So to kind of think along this line, what we did first is I want to first take a deeper look of what is the physical meaning of refractive index. Although we have been talking it a lot so far, but we actually didn't explain what is actually refractive index. So now it's 2 to 3 slides about the mathematical definition of refractive index. So it's actually pretty simple. So what is refractive index? Refractive index describes the interaction of electromagnetic wave with a certain material. So for example, here, electromagnetic waves, it has different color or different wavelengths, depending on which terminology you're more familiar with. And then for material, usually it has a quantity, physical property that describes its interaction with the light, that usually we use dielectric constant. And if you solve the propagation of light inside this dielectric environment, what you can get is the solution of a plane wave propagation inside this material. So you have this electric field and its propagation along one direction. And here we have this K, which is wave vector. And this wave vector is equal to square root of dielectric constant times the wave vector in the vacuum. I won't go to too much of the mathematics, but what is important here, you see the square root of dielectric constant. This is a mathematical definition of refractive index. So as many of that dielectric constant has usually when we talk about the dependence of dielectric constant on frequency, it has both real components and imaginary components. Similarly, because we're just doing a square root of dielectric constant, refractive index will also have real and imaginary components. Previously, we were just talking about, for example, water 1.33 is actually only talking about the real part of refractive index. A lot of time, people ignored the imaginary part of refractive index. So it is quite important to know this, because in physical quantities, a real and imaginary part has going to contribute different physical phenomena. Just a little bit introduction on what is this complex number representation. So a physical property, it can be representing to both real part and imaginary part. And I here is the imaginary unit. The square of the imaginary unit is equal to -1. You can also think about it on a complex plane where a physical property changes around this face. And then you can think of the magnitude, and here is the complex angle. And then you can use the Euler formula to decompose it into real and imaginary part. So if you utilize the Euler's formula and get back to our original solution for the wave propagation, and we can put this real and imaginary part of refractive index back to this solution. So what we will see is that the real part is going to determine what is the propagation speed of light inside the medium, and the imaginary part is going to contribute what is the absorption level of light inside the medium. So again, we start from light interaction with materials, and now we decompose two parts of the light propagation properties. One is relating to the propagation speed, the other is relating to the decay of intensity over this propagation. So very interesting fact is that you see we started from one physical property, which is this dielectric constant or refractive index, and it decomposed into two seemingly independent parameters, but actually they are not independent. When this phenomena happens, there's a mathematical rule which is called Cramer's chronic relation. This Cramer's chronic relation connects these two physical, real and imaginary part of the physical properties. Physically speaking, because this physical quantity is describing light interaction with the material. So this means that the real and the imaginary part, they are not independent. They are coming from the same origin. Utilizing some of the math causality rules, you can actually predict what's the connection between those two. And the equations are pretty complicated, but just briefly explaining this, what does it mean is, if you know all the imaginary components, you can back calculate the real components. If you know all the real components, you can also back calculate the imaginary components. And the equation looks very complicated. And here's a graph. So if we're introducing an arbitrary imaginary peak, and we can actually tune the real part in the nearby wavelengths. So what does that mean is that because imaginary is relating to the absorption of light, real part is usually relating to the speed of light, or usually what we call refractive index. So this seems to suggest by introducing material or absorbing molecule into something, some materials, we can actually tune the refractive index in a relatively easy way. And specifically, what we are interested in this wavelength, which is longer than this absorption peak, we find that the refractive index actually get increased. So maybe we can utilize this strategy to reduce the refractive index contrast inside the biological tissue. So let me just rephrase what we just said. So we start from the refractive index of water, which is about 1.33 across the visible to near infrared wavelength. And simply by introducing some absorbing molecules, for example, that absorbs around 400 nanometer, and we can tune the refractive index in the nearby wavelengths, the real part of refractive index. And part of the wavelengths actually the refractive index can be increased so that the difference between the solution with the high refractive index component inside our tissue, such as lipid and collagen, the contrast is getting smaller. So then the scattering is going to be reduced because of this reduced refractive index contrast. And importantly, what we find is that from this, we can see when the absorption actually already reduced to zero, this refractive index change is still remained. So that's important for us because we don't want all the light to be absorbed. We are actually utilizing a spectrum window where there's very minimum absorption of light. This theory or engineering thought looks very exciting, but it's actually pretty strict in terms of selecting these molecules, because we want molecules that are very efficient in terms of introducing these absorption peak, and we want it only absorb in certain wavelengths. And in other wavelengths, we want it to be spectrally clean, which means it doesn't absorb in other wavelengths. And ideally, because we are aiming for biomedical application, we hope that, excuse me, speaking too much. So we hope that
time to drink and drip if you want.
Yeah,
we have time.
Yeah, it's just every time I talk about this, getting very exciting. So we hope these molecules can be actually potentially biologically safe. So when I was doing this research in Stanford, when I was postdoc, I look at these selection criteria. And what came to me, it seems like we are selecting molecules with the same criteria with food industry. So, and at that point, I started to write to the largest food company in the US. Can you actually give me a few samples from food industry to test if it's going to work for my application? And then they send me something for free. And that actually leads to the most popular candidates that I'm going to share in the following. So our answer for these selection criteria is food dye, and specifically one of the most popular dye that we're currently using is what we call, and the industry name is actually yellow number 5. As we mentioned, this is already a chemical that is widely used in the industry. People make it in mass, very large quantities, like people sell it in kilograms, in tons, and they are very cheap, easy to acquire. And because it's designed for the food industry, you can prepare it very easily prepare it into different kind of aqueous solutions. And because they are designed to introduce color into the food, so they are very efficient in terms of adding the color. In our terminology, introducing absorption. And in other ways, and their spectrum are very, very clean. So for example, when we measure the absorption of these molecules at high concentrations, we can see that it absorbs at this peak, around this peak, but at a wavelength beyond 600 nanometer or further, it overlaps with the water absorption spectrum, which means that it doesn't actually absorb any light in these wavelengths at all. That's very important for imaging applications where most of our imaging is going to happen in these wavelengths. And lastly, because this is a food dye, it has been decently safe in the past years. It has been used in Doritos energy drinks a lot. Although recently there is new regulations since the end of last year. People hope to replace most of the synthetic dyes with more natural dyes. But anyway, it used to be safe, considered safe by food industry. So as I mentioned, we have this key concept of refractive index. So the important step for us is to quantify whether we can actually change the refractive index utilizing this food dye. So to measure the refractive index, what we come up with a strategy is what we use a tool that is called ellipsometer. So what this tool did is it shines a light from one direction and hits your sample and collects the refractive light. By analyzing the polarization change and intensity change over 2 different polarization directions, it can experimentally calculate both the real and imaginary components. So these are purely experimental methods of direct measurement of refractive index. These tools mostly used in the semiconductor industry to measure the film thickness, but here we are using it in a very unconventional way to quantify the solution refractive index. But utilizing this tool, what we can do is we can actually measure the imaginary part, which is this red curve. We can see this red curve actually corresponds to the absorption peak of this dye solution. And around this absorption peak, we can start to see how the refractive index actually gets changed in the nearby. And it indeed gets increased to a level that is comparable to some of the high refractive index components inside our biological tissue. And as many of you may be familiar with that, you can tune the absorption of a solution simply by tuning the concentration. So these also gave us a way that we can actually tune the real part of refractive index by tuning the concentration as well. And then as you can imagine, for different components, if we want to reach a different refractive index target, we can easily tune that by changing the concentration of the chemicals And we can, for example, at different wavelengths, it will have different dependence. If we want to achieve a certain level, we can further decide what is the optimized concentration for this application. Okay, so far we find this chemical, it actually changes the refractive index based on what we hope. And does it really work for biological tissue? So now here's some results that my postdoc, Wakas Shabir, did in our lab. So for example, this is a piece of chicken breast tissue got it from supermarket. So you slice it into a few millimeters thick. So this is about 2 millimeters thick. Because of the strong scattering inside the biological tissue, when you place this on the grid, you are not able to see the pattern below. So this is kind of matching with your intuition of how you expect for tissue you got from the supermarket. However, after you soak this tissue into this solution, you can see that this tissue becomes transparent, and this allows you to visualize the patterns below. And I usually take a second for you to emphasize that this is actually a quite counterintuitive experiment, because let's just think about what we have. We start from a tissue which is optically opaque, and we are introducing it into a system where it's a solution which is strongly absorbing. When you mix them together, it is very hard to expect that these two actually, you can make it transparent, because both of them seems to have some adverse effect in terms of light propagation. But when you mix them together, they actually become transparent in certain wavelengths. So that's the beauty of physics. If you know the physics, things seem simple. But if you don't know physics, this is more like a magic trick. So let me just do a little bit more advertise. You can hide anything. So you can hide our logo, and you can also see through this tissue as well. So more quantitatively speaking, so we can also use spectrometer to actually quantify what is the level change in transparency between these two tissues. So as I mentioned, the tissue is opaque at the beginning. Quantitatively speaking, from a optics perspective, it means that it blocks the light. So you can see from visible to near infrared, the transmittance is very low, usually approximately 0 or at most 5% in this very broad wavelength range. But after we treat it with this dye solution, we increase the transmittance to like 30 to 40%. And especially in some of the near infrared, we can even make it even getting close to 40%. So this is just a quantitative explanation of how we actually make the tissue more transparent. These experiments are super easy. So here's a video I'd like to show. So this is a tissue. And once I click the start, we are pouring the solution inside. As you can see, about 10 minutes, the tissue is gone. So as you can see, it becomes very hard to actually now tell where are the tissue anymore. So because this is such an easy experiment, after our work gets published, I got a lot of emails from students, high school students all over the world who want to reproduce this experiment in their garage. Because everything is cheap. You got tissue from supermarket, you can get these food dyes. They are also available to everyone. It's not like restricted chemicals. So the students write to me how to reproduce this and can they write a term paper about this project. So these days I'm also hoping to design some outreach activities to show people how these experiments actually work. So again, going back to science, is that because this is relatively simple, immediately after our preliminary demonstration, we were thinking, can we utilize this actually for live animals? So as I mentioned from the beginning, that light blocks our tissue usually blocks the light because of this refractive index. And now we make it more uniform. Light can go through to the detector without much scattering. And because it seems like this method can be easily translated towards live animals. So one challenge we face is how are we going to introduce to live animals? So the strategy we came up with is we actually prepare a soft gel. We add a little bit of into the solution to increase the viscosity so that it's more like a cream. So we learn a lot from cosmetic industry these days. So how to actually apply these molecules onto the animal skin. So we prepare a soft gel and apply to the animals to whatever location we want. For example, firstly, let me first show you, this is a mouse head. After you shave the fur, this is how it looks like. Because of the strong scattering in the skin, we are not able to see any feature below it. And we then apply this dye molecule on top of the skin, and we can make part of this skin transparent. And then this allows us to directly visualize the vessels on the surface of the brain. So if you remember at the beginning when I was talking about this invisible man, one challenge he had is he failed to reverse the transparency. In our case, because this is a physical method, what you can do is you can wash these molecules away and then you will see the tissue become opaque again. So it's just, we don't have the challenge of making it reversible. So this is just the images taken by our phone or what we see by our eye. If we want to have a better contrast, we can also use laser speckle imaging. It's a method that to specifically create contrast for vessels, just in a short explanation. So what they do is shine a laser onto these features and then it will pick up features which are corresponding to vessels. So here are the illustration. Before we apply any of the transparent dye, we cannot see anything below this because of the strong scattering of the skin. But after we make the skin transparent, we can actually directly visualize the vessels on the brain. So just to confirm, these vessels are actually on the surface of the brain instead of in the skin. So what we do is we actually remove the skin and these are directly look at it so we can compare the features and confirm these features are indeed on the surface of brain instead of in the skin. So it's a very versatile tool. So for example, another example where I'm showing here is that if I'm interested in looking at the muscles in the mouse leg, what I do is I can apply this dye and make part of the mouse leg transparent. And these allow me to directly visualize the muscle bands inside the mouse leg. And these are actually very high resolution imaging, relatively high resolution imaging, because this individual bands is about 2.5 micron. So here we are trying to show that this method can be compatible with a lot of existing optical imaging modalities. So by making this transparent, as you guys, here is a video where we can see we can do a three-dimensional scan. At the beginning, we're seeing skin features, and then when we go deeper and deeper, you start to see these vessels below it. So, right. So in the past, what would be when people are interested in studying the muscles, what they have to do is they have to poke a needle, or what we call a gray lens, a needle-like lens, into the muscle to look at this. But now we have a method. We just apply this gel, and then you can image this directly. So another demonstration what we want to do is we want to actually make this more obvious to everyone. So what we do is we apply this dye molecule onto the belly of the mice. And because of the strong scattering of the skin, at the beginning, we cannot see much. But after we apply these dye molecules, we start to see the contour of the organs. And my student, David, actually, he's a student from electrical computer engineering. He utilized some of the image processing tools. We can increase the contrast of the vessels and of the organs. So now it's like we can directly, using optical method to visualize, examine the internal organ dynamics. So again, this method is completely reversible. So this is an illustration that at the beginning, after we make it transparent and we can wash it away, and then we return the animals. So we are not going to sacrifice the animals. That's another selling point of this method is we return the animal back to the animal facility, and we can see that over several days, the fur are gradually growing back, so that you can continue working with the same animal over an extended period of time. And we also quantified how long it takes for this food dye to actually from the animal body. So it usually by tracking the urine and feces, it usually takes about a day or two. Yeah, I think I need to go a little bit faster now. So as we mentioned, although we are focusing on one of the chemical here, by changing the absorption of the molecules, actually it opens a lot of opportunities. And for example, if you don't like this yellow color, you want it more transparent, we can actually give you another option where the transparency is more close to visible color. So in this case, you can actually see the internal organs and directly compare with more like a textbook anatomy of the mice. So you can see liver, small intestine, cecum, bladder. So why this is important is that we are not only seeing that, it's actually seeing it in live animals. For example, now we can see the mouse are still breathing, and then you can see the internal organs from all different angles. And we can also see how the organs are actually move inside our body. Right. So this is a very important question. And in the past, just a few examples how people are trying to study this in the past. They either have to take the internal organs out and study it under a microscope, or they replace a large part of the tissue with transparent window to visualize the internal movement of the organs. But now we are giving an alternative method that seems to be a lot less invasive at this point. So again, this is a very important question: how food actually propagate inside our body, because gut controls the absorption, excretion of food, and movement of these substances from one direction to the other. So in biology, these are actually realized by a sophisticated nervous system that is called enteric nervous system. These neurons are embedded in the muscles of the nerve, in the muscles of the gut, and they control which part of the gut should contract, which part should relax, so that food are propagated, pushed from one direction to the other. It's not going to mess around. So when I was at Stanford, I had a co-advisor, Professor Julia Kochmitz, and through her guidance, I learned some genetic tools. How can we utilize genetic tools to label the neurons inside the gut? So here's an image that typically people do in the past. They took the gut out and took an image under confocal microscope, and you can see these neural networks. But now we are proposing a method that we can just make part of the gut transparent. And these allow us to directly visualize these networks inside the live animals. And importantly, we are also offering something that people have never seen before, is how these networks are actually moved inside the body without taking it out. So based on these results, we started to discover some of the things that people haven't discovered before, is how these networks are actually moving around. For example, we find that they actually periodically move around, and there are bursts of movements inside these networks as well, movement as well. And we also developed some computer analysis methods to actually visualize the complex motion inside these neural networks. There could be local contraction, local expansion, local rotation. These are all information people don't know before, and hopefully we can connect these kind of characterizations with future diagnostic tools so that we can learn what are these actually mean in terms of their physiology. Right, a little bit more about what this works. So since this work is published, it was at the end of last year, it was ranked as the top 10 breakthrough in physics in 2024. And you can see this is me. I'm holding a dye solution, and these are actually food dye directly extracted from the snacks, which is the thing I mentioned at the very beginning. So it got a lot of media attention. We are particularly excited to see how broad audience, like the public, got interested in our work. There are probably over 260 news report about this work. So this is from CNN, and this is the Times, and that's why I got an inspiration. And because they are relating our work with the famous novel of The Invisible Man. So that's not my original idea. I actually ran from them. But more importantly is that the whole biomedical imaging field is getting very excited, because now from their perspective, we have a potential method that we can make mouse transparent. Maybe not like this. These are, AI can do whatever they want, but we can make part of the animal transparent so that it can do a lot of important biomedical discoveries. So immediately our work is released, some groups started to adapt this method for their own applications. So they first reproduced how they actually visualize the internal organ, but they also do something beyond my original paper, is that this group, they are interested in spine, spleen, so they actually visualized features I wasn't familiar with before. And because from our perspective, one I usually advertise is that our method can improve any of the existing optical imaging modalities. It sounds very bold, but this is what we see in the past year or so, is that people are starting to using these molecules to improve the imaging capabilities of optical coherence tomography. So here you can see these are comparison of without ultrasound and with ultrasound. Your imaging depths and resolution get improved a lot. And people are also showing this to improve optical photoacoustic microscopy, and then they are utilizing this to visualize the brain. And by applying these ultrasound molecules, you are starting to see a lot of features, deeper features that you are not able to observe before. Apart from that, very recently, I'm very happy to see that, because as I mentioned, it's not just we are reporting ultrasound itself. Is that the importance of our technique is we actually reported a framework starting from first principle. How can we find molecules that can lead to making tissue transparent. And we can see that these are very recent work, I think just published in September. Yeah, so they September 1st. So they are actually reporting that indocyanine grain is another approved dye molecule used in clinic. And by utilizing these dye molecules, they can improve the optical coherence tomography results in the skin. And this is a comparison, and this is also reversible. And what is also particularly exciting to me is that there's another preprint utilizing a molecule called fluorescein. And what they're showing is that by utilizing these dye molecules together with surgery, this can allow doctors to identify features that they were not able to see before. And this will help the doctor to make more precise incision, more precise cut. And I feel like our method actually has a lot of potential in the future of biomedical science. So this is an illustration NSF made for our technology because it was partly funded by NSF when we were doing this. And I really hope that in the future is that whenever you want to inspect part of our body, we just apply these kind of absorbing molecules and then look at it and then take a photo and send to a doctor. So you don't need to go to hospitals anymore. So that was my hope for this technology. So at last, I would like to conclude with a very interesting series. So one of the comic company in the UK, they got inspired by our work and decided to draw a series of comics based on our discovery. So you can see this person is eating Doritos and rub it on the belly, and his belly become transparent. And then he actually made a series. I won't go through all of that, but I feel like what is particularly exciting for me is that the general audience, the broad public, got excited in science, and more young researchers are joining this field, which I feel is very much rewarding as a scientist. Lastly, let me acknowledge my group. So it's a very pretty much, as you hear from my talk, we use IME physics, but we also work with chemistry, biology, computer engineering as well. So it's a very diverse group, large number of people. And we also collaborate a lot with people in physics, neuroscience, bioengineering as well. I'm in Dallas, so we have very close connection with UT Southwestern, which helps me to further expand the biomedical applications of this technology. Last, I have some funding support from Kavli Foundation and also funding from UTD. And we acknowledge all the contributions from our favorite mice and really appreciate their contributions. Thank you.
Thank you so much. That was fantastic. Really, really extraordinary. Also, it's cute that you're thinking your favorite mice, because I assume that they might also still be around because you actually can reuse them.
Yeah.
Okay,
Lovely,
amazing.
lovely.
Well, I mean, yeah, this was, it really looked like magic to me. So thanks a lot. You really rocked my brain here this morning. Micah already has a question. I should also remind everyone else, if you have any questions, comments, or like follow on comments, please raise your hand, or if you can't raise your hand, just put it in the chat and I will read it out for you. And we will start with Micah.
So I attend a lot of foresight talks, so I'm largely desensitized to cool science. But this presentation legitimately excited me. It was the first time in a while. So good job on that. I have a ton of questions, but I'm just going to ask just one so other people can ask as well. Can you mix different molecules in like just the right ratios to expand the spectrum of visible light that goes through without refracting, or does that make this problem worse?
So we can potentially mix them. But one thing we need to be very careful is when you mix different dyes, we have to make sure that there are certain wavelengths that are not absorbing or imaging. So a lot of times when you're mixing different dyes, for example, it may absorb somewhere here, and then it kind of compromise with the low absorption for one dye. So they have to be so that there's a region of this in terms of spectrum that are not absorbing for application modes. Yep.
Oh, you're getting a thumbs up there. I'm sure Micah has more questions, but let's start with Ted after that. Ted, go ahead.
I like to also for that was a very fun talk, and I think very accessible.
Thank you.
How you describe what the physics are and also the results. So my question, I was wondering, would it help in terms of like the resolution or the focus on things you could get if you could more precisely position where the dye is inside the animal or in parts of the skin. You described a cream that goes over as far as like the whole belly or whatever. But if I'm thinking things maybe like nanoparticles, things that could bind the dye to exactly where you would want it in some structure.
Right.
Like that kind of positioning control. Would that be helpful?
Yeah. That will definitely helpful. That's one of the direction we are hoping to actually address, because this is if we go back to the very beginning when we are talking about this heterogeneity of refractive index inside the tissue, it would definitely be better if we can be more accurate how these molecules actually can be targeted towards certain features that has high refractive index. And unfortunately, currently, we don't have a very good way to achieve this yet. I think at this point, we are just relying on passive diffusion of the molecules through all the tissue. And apparently, there's a limit we are going to hit, because this uniform change of refractive is not going to be as good as if we have a more optimized way of delivery to a certain target. But that's something we are actively doing.
Yeah, and I was saying also, because you mentioned toward the end having different dye molecules, and you might have different ones in different locations depending on the
Yeah. But at this point, what they are, although it's different molecules, but we are all still relying on passive diffusion and others as well. But passive diffusion is going to have a limit. That's something actually we are going to share with the community very soon, that this is not we have to come up with new strategy. How can we be more accurate to
this is wonderful work.
break this? Thank you. Yeah. Thank you.
Yeah, really cool. Michael, you're up next again.
Yeah, this kind of builds off of previous Ted's question.
Right.
Does the dye, when you apply it as a cream, does it actually get into the cells, or does it just kind of get into the tissue between cells?
Right. Wonderful question. So it's a very hard, I think we believe it is getting everywhere into the aqueous phase of the tissue, but mostly in the interstitial. We did some ex vivo test, but we see that it's going everywhere, but it's not a conclusive results yet. We are hoping to do a more higher resolution in vivo experiment to see actually it go across the membrane or not. Because we did some in vitro test, so it's not the exact system. We basically incubate the cells with the dye molecules. So we are seeing that it's not easy for the dye to cross the cell membrane. But in some of the samples, when we are doing ex vivo test, in vivo test, and then we sacrifice the tissue for that, we see that the dyes actually go into the tissue. So, but there's some debates over this question that we are trying to clarify further. But that's a point that we are also hoping to address in the future.
And the one where you did the scalp, do you believe that it got into the bone as well, and you're able to see through the bone, or were you only seeing just like on the surface?
Right. So what we believe is it's mostly clear in the scalp, and the bone is more like a layer that is protecting the dye diffusing into the brain. That's what we believe at this point. The natural question is why then the bone is here, why it's not interfering with the light. In this case, because we are working with mice, which the bone is not that thick, the bone is less than 0.5 millimeter. So actually, especially when they are still in the interstitial fluid environment, the scattering of the bone is not that's kind of challenging for this microscopic imaging. However, for some of the high resolution imaging, it becomes more problematic.
Gotcha. Have you looked into transparent organisms at all to see if we can learn anything or figure out what chemicals they do? And do we know, are they using the same technique, or do they do something totally different?
Yeah, wonderful question. So we have a collaborator here at UT Dallas, and he is actually working with zebrafish. So a lot of the existing work working with this transparent organism, they try to identify this species, which is apparently more transparent, at least in the young ages. So most of these transparent animals, they are aqueous species. When we try to combine this with our method, it's a challenge because it's not like mice. We apply this, it stays in the animals. When it's an aqueous species, this dye goes everywhere. So, that becomes a challenge of combining this with the existing transparent species study. So we haven't come up with a strategy how to go over that yet.
Okay. And I swear these questions will end eventually, only a couple more. Have you thought at all about figuring out a way to make a transgenic mouse that constitutively produces something equivalent to yellow number 5 in every tissue, in all the cells, just universally. And like, what are your thoughts on that as just a way forward to get a fully transparent lab mouse that we could do research with?
Yeah, so that's one of our final dream, that one day we can produce a mouse line that has certain transparency recorded into the animal directly. At this point, we don't, because I didn't emphasize much on this. The concentration of the dye itself is still very high, like hundreds of millimolar level. That's too much for typical biomolecules inside physiological state. What we are trying to do right at this moment is try to identify more effective molecules so that we can lower the dosage rate, and then when we can lower it to a level that is comparable with, for example, protein expression level inside the mouse or other organisms, and then we can probably combine this to develop certain feasible models. That's our plan. But I feel like hopefully this is a much more challenging task. I would expect like probably 5 to 10 years, then maybe we can have some preliminary results to share.
Amazing. I know we're very short on time here, so and I hope it's the correct pronunciation of your name. If you want to sneak in a question.
Yeah, Nias.
Yeah.
Hello, can you hear me?
Yeah, again.
Oh yeah, super nice talk. Really enjoyed.
Yeah.
I was wondering about the structure of the tartar scene. It looks like it's a photo switch because it has an azo and then a double bond azo. So do you see any switching? Because if I think about other switching molecules, the absorption should change when they switch. And do you see an effect on refractive index due to switching?
We haven't done this, but I think your thought is very inspiring, actually. Yeah.
I
think that's definitely doable.
Yeah.
If you can tune the absorption utilizing a switchable molecule, then possibly you can tune the transparency as well. But we haven't done that. Yeah.
Okay. Yeah. Looking forward.
Yeah. Thank you. Yeah.
Any other questions from the audience? Otherwise, I'll get a few more general. Yeah, Micah, go for it.
I have one more that I just thought of.
Yeah.
Could we engineer proteins that do this, or does this effect limited to small molecules? Like, does the molecule itself need to be generally pretty small in order for this to work?
Yeah, protein can work as well. Yeah, I think that it definitely can work. It's just a little bit, proteins are more expensive when you need a very relatively high concentration. It's generally more expensive. The other point is that you have to design the protein in a way that it absorbs more close to the visible wavelength. A lot of proteins, if they are absorbing very far away from this wavelength, then you have to use a lot of molecules to achieve this effect. So that's a short answer. So if you want the protein to be efficient, you want to bring the absorption more close to the visible wavelength.
Gotcha. I was just thinking if you can get the cost of testing down significantly, you can just generate huge volumes of data and then train an AI model on to learn how to predict the absorption of any given protein, and then just ask it, give me some proteins that I can test and that absorb the light at the wavelengths we want. And then with proteins, then it becomes much easier to generate transgenic mouse.
Exactly. Yeah, the thought is totally correct. It's something we really hope to do, but it's a little bit beyond my expertise.
All right, let's see what I can do in a few minutes. I'm mostly asking the kind of like speculative questions at the end of it. You mentioned, I think, that perhaps we won't be needing to go to hospitals anymore. Of course, that was like big goal. But if you were to put on your speculative head, like where could this be in like 5 to 10 years? Like what kind of like actual applications could we see that can benefit humans, other living species all around?
Yeah, so I'm glad you asked this. So why I was really hope to bring it to human is that I have this life experience when I was during COVID. I hurt my ankle during play tennis with my friend. And then after that, I have to take an x-ray. It was during COVID, and then no one wants to see each other. So I have to struggle to hospital and take an x-ray. At that point, I was dreaming that if my technology one day can go to everyone's home, and then I can just take a photo at home and then send to doctor. So it's actually will make our life a lot easier. I think that's why I feel that was one of my vision for the future of this technology. it has a lot of possibilities in terms of research field. I think that's definitely true, like all these transgenic models. But I think these impacts are more limited to academia. But academia, we really try to push towards clinical demonstrations, like for early disease diagnosis, health monitor. We want to combine this with existing tools that can enable us to detect earlier and sense some of the biomarkers that typically we're not able to access without this technology.
Okay, awesome.
Mm.
Any final words on how people can find you, where, what's next for you in that work, and how can people follow along? Because this was amazing. I'm very appreciative of your work.
Yeah, we are continuing developing this. Our group is a pretty diverse group. People work on optics, working on chemistry, how to actually improve this further. And it's always easy to either connect on LinkedIn or just visit our group website, because we keep everything updated on our website. What are we doing? What are we sharing with the community at this moment?
Fantastic. Thank you so much on behalf of the group. Thanks everyone for your very fun questions. This was, this is a
Glad
great one.
liked it.
And I look forward to hopefully seeing you soon when you've eventually managed to do this with an entire human.
Yeah. I'll see you later this year, I guess.
Latest in person. Yeah, I'm excited. Thanks everyone. Have a good one.